far we've talked about how cells are structured and how they divide. Now we're going to be looking at something just as important, how substances move in and out of cells. Because if you think about it, cells are constantly taking in useful substances and removing waste. For example, organisms need nutrients to survive. Some organisms, like plants, are autotrophs, meaning they make their own food using carbon dioxide and water through photosynthesis. Others, like humans, are heterotrophs, meaning they obtain their energy by consuming other organisms. At the same time, cells are constantly producing waste products through metabolic reactions. If these waste products build up, they can become toxic. For example, carbon dioxide is produced during respiration and needs to be removed from the cells and transported to the lungs. Similarly, urea is a waste product that must be transported out of the cells and eventually excreted. So this raises the key question: how do substances actually move in and out of cells? To answer that, we first need to understand the cell membrane. The cell membrane is made out of a phospholipid bilayer. Each phospholipid has a hydrophilic head and a hydrophobic tail. Hydrophilic meaning water-loving, hydrophobic meaning water-hating. This structure makes the membrane semi-permeable, meaning it controls what can enter and leave the cell. Some substances can pass through easily while others cannot. So substances move across this membrane in two main ways: passive transport and active transport. Let's start with passive transport. This includes diffusion, where particles move from an area of high concentration to low concentration. This happens naturally and does not require energy. Now, osmosis is a special type of diffusion, and this is where things can get a bit confusing. So let's break it down using an example. Imagine you have two identical cells, red blood cells, and you place them in two different solutions. First, you place a cell into a very salty solution. This is called a hypertonic solution, meaning it has a very high solute concentration. And because there's so much salt outside the cell, and there is less water outside compared to the inside of the cell. So what happens is the water moves out of the cell into the solution. As a result, the cell shrinks or shrivels up. Now let's look at the opposite situation. You place a cell into a solution with very, very little salt. This is called a hypotonic solution because it's got a lower solute concentration. This time, there is more water outside the cell than inside. And water is always going to move to the place where there isn't as much water. So here, the water is going to move into the cell because that's where there's less water. And this is going to cause the cell to swell, and in extreme cases, the cell might even burst. So the key idea with osmosis is that water moves from an area of high water concentration to low water concentration, or in other words, towards the area with a higher solute or salt concentration. And because this movement happens down a concentration gradient and does not require energy, osmosis is a form of passive transport. Now let's briefly look at active transport. Active transport is different because it moves substances against the concentration gradient, from low concentration to high concentration. So this requires energy, ATP, and is usually carried out by protein pumps in the membrane. There are also other methods of transport like endocytosis and exocytosis, where the cell membrane forms vesicles to move substances in and out in bulk. Endocytosis is moving in, exocytosis is moving out. Finally, there are a few important factors that affect how quickly substances move in and out of cells. One key factor is the surface area to volume ratio. Smaller cells have a larger surface area relative to their volume, which allows substances to move in and out more efficiently. Another factor is the concentration gradient. The greater the difference in concentration, the faster diffusion occurs. So overall, the movement of substances in and out of a cell is essential for survival, allowing cells to take in what they need and remove what they don't.